Hi everyone, as Grandpa Miner said, my name is Zach Meharg. I am a fifth-year PhD candidate at Auburn University, down in Alabama, but not at Auburn, and also HudsonAlpha Institute for Biotechnology in Huntsville, Alabama. So, in case you don't know where Huntsville is, I got to drive this very long route on Thursday because I drove here from Huntsville because it saved me a little bit on gas and everything. But this is HudsonAlpha. It was essentially how this HudsonAlpha started is in about 2010. A couple of the lead researchers for the Human Genome Project, they finished a genome project and they're like, we want to start a company. Where do we build this company? You go where the land is the cheapest, which happened to be in Huntsville, Alabama. So they moved to this, I don't even know the exact size, but we moved to this wonderful building. If you're ever in Huntsville, I highly recommend going. It has the world's largest DNA walking trail, which is a 1.5-mile trail where you can walk both strands of DNA, where they have a bunch of different facts about the human genomes and like where particular diseases are on those genomes. One of the main reasons why I drove here is I attempted to grow dahlias at my house this summer, which they did not do the greatest. I will say, granted, this is after they were moved because I moved houses about 2 weeks ago. So they survived them. They were looking good, and then we got into the heat of Alabama summer, where it is very humid, and I think the average was about like 90 to 92, and we're at normally on a good day like 70% humidity. Thankfully, I was able to get. I had one flower that survived, and I was gonna bring it. It's actually still in my car. So when I was driving here, I looked down 2 hours into the trip, and it had lost a couple petals. So it is still staying in my car. Yep. So now I'm done with a little bit of that. I'm going to go into the background of my talk. Did it cut off? I apparently spelled my last name wrong, but that's okay. So I always like to show in any of my talks, whether or not it's to here or to local societies or even to other researchers, other genetic researchers around the world. I always like to show this talk because when I talk about dahlias, people are like, oh, that's a flower. But none of them really understand how much phenotypic diversity is there in cultivated dahlia. Obviously, all of you do, because that's why you grow them. But when I see this picture, and every time I show this, I always hear awes, especially from people at HudsonAlpha, and just how beautiful they are on the outside. But I think I'm lucky enough to also realize how beautiful they are on the inside, partially because I've been having to look at the inside of dahlias, the DNA of dahlias for the past 5 years. Why? Oh, okay. Yeah, so dahlias are also beautiful on the inside. And the answer to dahlia's beauty is packaged in a varying number of chromosomes. So here I just have an image that was taken in 1920, where all of these little dots are all chromosomes of various different species, and we have Dahlia variabilis, which is cultivated dahlia, that has 64 chromosomes. We have a couple different species that have 36, some that have 32, and some that have 34 that aren't pictured here and on here. But cultivated dahlia has 64 chromosomes, and the way that these chromosomes have been packaged has been up for debate in literature. So if you Google how many the main way that I think about this is we talk a lot about polyploidy in dahlia. And there's two camps of dahlia research. There's dahlia as a tetraploid versus dahlia as an octoploid. So the main way to look at this is basically how many groupings of chromosomes are there. So humans, we're a diploid, so we have one copy from mom, one copy from dad. When we look for tetraploids, we would expect to see 2 copies from mom, 2 copies from dad, making 4 copies total. And if we look for an octoploid, we would look to see 4 copies from mom, 4 copies for dad. So some examples of octoploids that you might not, or you may not know as an octoploid would be like strawberries. The strawberries that you'll buy at the store are all octoploids, and for tetraploids, a good example that would be blueberries, but in some cases there's some funkiness going on. But so, that's the wrong button. Okay, so the real question is, why is this important? And so when we're trying to build a genome, which is what my whole first aim of my dissertation is, it's a lot easier to build a puzzle. I like to equate dahlia to a puzzle. So basically, you have this box on the table, and you open up the box. I'd be easier to assemble a puzzle or the box if there was only 4 copies of that puzzle in there instead of trying to assemble 8 copies of it. And so really, one of the only ways to figure that out is to actually start going out and building a genome for a cultivar. Which leads to what cultivar do we pick to build a genome for? That's one of the nice things about working with the ADS is we talked about what criteria do we want to be to do a good cultivar for. Because at the time there was no Dahlia genome out there. I apologize for the formatting. It was a little bit different when I was doing this, but it says the main criteria we wanted for a good genome would be it's been around for a long period of time, it still continues to win on the show floor, and it is significant to the American Dahlia Society. And I happened to walk around, and I was happy that there's still one of them out there today on the floor, and that happened to be Edna C. Because Edna C was introduced in 1968, it still continues to win on the competition floor. I believe last year it even won a couple blue ribbons. And that in 2015, it was voted one of the best Dahlias in the last 50 years. And so from here, we have to extract the DNA, and it's it's a good segue to go from the virus talk, where we talked about the virus being 8,000 base pairs, and then the human genome being 32 billion, or 3.2 billion base pairs. The Dahlia genome is roughly 8 billion base pairs, so almost 3 times the size of the human genome, which makes it a little more complicated, and why we're hoping to find only 4 copies instead of 8 of a number of chromosomes. So to do this, we do a bunch of DNA extractions, we do a bunch of sequencing. I bang my head against the wall a couple times because you run a program and you think you give it enough memory, and then you find out after 20 days of running that you didn't, and then you have to run it again. But those are just tiny things that end up leading to so that we can create this very checkered board-like image over here, which is what is called a contact map in the scope of genome assembly. And essentially all this is it lets you visualize what your chromosomes look like and how many chromosomes are there. And so for this particular puzzle, going on that analogy, I started with about 4,781 pieces of the puzzle, that some can be up to 100 megabases, or about 100 million base pairs long, so A, T, C's, and G's. And some of these being roughly about 100 base pairs. And so you're trying to figure out where each of these pieces go, because each of them has a correct place to go, but it's very hard. You're not necessarily which way to go. And thankfully, a lot of the tools that are out, or unfortunately in some cases, a lot of the tools that are out there were designed to work on the human genome or much smaller plant genomes. So ones that shouldn't take it too long to run. Unfortunately, that's not the case for Dahlia. So it took a couple months just to get to this stage, and then it took another few months to actually go through and manually build each of these boxes, because each of these blue boxes is one Dahlia chromosome, and then inside these blue boxes that I have turned off would be a bunch of smaller boxes of the individual pieces that got put together. So in puzzle analogy still, this would be sort of like each chromosome would be like sort of grouping your colors together based off what your picture is, and then it's trying to figure out where that particular group of puzzle pieces needs to go in the whole grand scheme of the picture. But the big thing to take away from this, which is kind of hard to see here, because otherwise, like if I look at this and I'd be like, oh, I know exactly what I'm supposed to see here, because I've been looking at it for so long. But what we want to see, what we were looking for here is how many groups of each chromosome was there. So if you look here at these four blue boxes, we have 4 lines from 3 other chromosomes that help to tell me that those 4 chromosomes are all the same thing, and that we don't have any other ones from another group that say, oh, I'm also the same thing. So this would indicate that cultivated Dahlia, at least for Edna C that we assembled, is a tetraploid and not an octoploid. So making it a little less complex than what we were expecting when we first started. Since I started this project 5 years ago, another Dahlia genome came out for Calvin Floodlight. So Edna C isn't the only Dahlia genome out there. And we do some comparative work, which is what I have here. We can see that one group of 4 Edna C chromosomes corresponds to 4 Calvin Floodlight chromosomes, also helping us to show that at least in this case with Ednocene and Cultivate, that they're both tetraploids. So kind of, we can sort of start going away from the fact that Dahlia is octoploid, because at least in the 2 cases of genomes that are out there, they're tetraploids. That's not saying that there aren't octoploids out there, but it's just that the ones that we've looked at so far are all tetraploids. And so that kind of wraps up one chapter, and initially the first goal of this project was to just build a Dahlia genome. But as genetics has become more advanced in the field of genetics and genomics, that doesn't really bode well for a dissertation. That's more one chapter. So then I had to start thinking, what else can I be doing with the Dahlia genome in order to build a dissertation and hopefully eventually a career around it? And so I just wanted to put this out here that a genome is just the starting point for all genetic research. For example, the Human Genome Project was finished in 2003, and even to this day, people are still coming out with new experiments that they're trying to test on humans, or not on humans, on the genomes of humans, and trying to identify the causes for certain diseases by the genome. Because initially, when the Human Genome Project was founded and started, their goal was to, by having a genome, to be able to solve all the human diseases out there. And they quickly learned after that that's not necessarily the case. So I think even to this day, we're on like the 5th or 6th iteration of the human genome, and thousands of papers where people are using that genome to actually identify disease variants and causal genes for why people have certain hair color and stuff like that. So this leads me to so let me be thinking, what is the next thing for Dahlia that I could do? Really, the possibilities are endless. It kind of just depends on the experimental setup and also the funding for it and the money that you can get. So the first one that I thought of was, is it possible to identify what traits cause your favorite Dahlia? The next one that I thought of is, can we introduce new traits from other related species of Dahlias? So we're going to talk about the first one first, which again, I apologize for the formatting, it got a little bit off. But when we want to try and identify traits, the best way that we can do that is to create what's called a mutant library, which a lot of people will do for large crops. Dahlia is a great example of a time when we don't have to do that, because we're able to source a large diversity of flowers all in one place. So these are just a couple of the pictures that I could take where you could actually see the flowers this afternoon and not people's heads. So when just walking into this room, all of you see different cultivars, but I see a couple of base pairs that got changed in a couple of base pair changes between all of these ball Dahlias, which lead them to being all the different colors. Or even some examples that could be from a ball Dahlia to a stellar Dahlia. And so to do this, we need to actually sequence all of them, which we did in the 2021 show. And so I just want to iterate that all of you grow and assemble probably one of the most diverse collections every year that I've seen and anyone in my lab has seen for trying to do these large-scale genotyping soft programs. And so from the 2021 show, this is kind of a breakdown of the flower size on the left and the colors on the right, but the colors of each of these corresponds to more of the floral form compared to the actual color. But from here, we're able to do some cool groupings where we can say, okay, I want to look at all of the Dahlia cultivars that make more than one row of ray florets. Or I want to look at my ones that only make one row of ray florets. So it'd be like your anemones or your singles, and your anemones that only have 8 ray florets, your collarets, and your orchids and stuff like that. So with there, you're able to create something called do a genome-wide association study, which again is only possible if once we have the genome actually done. Because basically what you do is, and what we did is we sequenced 370 cultivars from the show at roughly 3x coverage. So we had about 24 gigabases of data for each of these cultivars. And then you take it and you put it all out in a line against your genome, and you're basically playing a spot the difference to see what's different between your genome and what's different between that particular cultivar or that group of cultivars, depending on what phenotype you're looking at. So for this example, I looked at I basically took out all of the Dahlias that were singles, anemones, orchids, collarets, anything that had less than anything that had 8 or less ray florets, and said, okay, what can we find any genes that could tell us, okay, when a dahlia has this specific gene, it's going to be more likely to have multiple rows of ray florets. And so this is called a Manhattan plot, particularly because when you're doing large number of samples in this sort of study, typically people do like thousands and thousands of samples, you'll tend to see higher lines or dots that come up way up here, and it's supposed to resemble the Manhattan skyline. This road doesn't really seem to resemble that. It feels like more like like Kansas. But the important thing to see here, which is nicely drawn out by these dotted lines, these solid lines, is that these lines are indicated by a couple different statistical tests that this particular, wherever this line is called a SNP, or a single nucleotide polymorphism, which is a big word for essentially just meaning if you have an A in one spot, you can go to a T, a C, or a G. That's all that stands for. And so what this is saying is that in these 4 regions, that's potential candidates that we're going to have to go in and explore to see what genes are nearby these potential SNPs, and if they have an effect on flower symmetry or meristem identification. And so we're sort of diving into those net results right now, but then I have you thinking, what other genes can we find with this? And so, for example, we could look at floral color, what makes a white dahlia white, what makes a red dahlia red. Additionally, look at more floral forms, like what makes a ball dahlia different than a water lily. Potentially, you could look at things like scent, because I know some dahlias sometimes have a scent to them. You could look for more cut flower industry of cut stem longevity. You could look for things like more blooms per plant. You could look for things from like tuber production per plant. And eventually you can even start to look for disease and viral resistance. And there's so many more. The possibilities are endless. Just if you need to think of a good experimental design, and you can figure out whatever genes you want. So from here, it led me into thinking the second step that we talked about is, can we introduce new traits from related species? So on the right, we have Edna C, and then on the left, we have a couple different species dahlias that I grew at our greenhouse. So we have a Sorensenii up here on the, Sorensenii up here on the top, a Coccinia down here, and then 2 different Rutuses that are siblings that I thought I would just throw in here as well. So in order for us to actually try and identify what species could be good sources for good genetic material into our cultivars, because I know there's examples in other crops where people will find a closely related species that has a disease resistance trait or a better berry trait, and they'll breed it into their cultivated crop, and you can create something called hybrid vigor, where you get bigger flowers, better blooms, more healthier plants. But in order to actually do that, we need to know how closely related cultivated dahlia is to some of these other species. Because you don't want to take a dahlia that is a species that is very far diverged from cultivated dahlia and try and cross it in, because the likelihood of them actually being able to create a viable cross is pretty low. To do this, we need to look at where do we source material from. So here is a map, and where, at least based on iNaturalist results, where all of these dahlia species are located. And for the most part, they're all primarily located in central Mexico. And so with this knowledge, I knew that I wouldn't be able to go to Mexico to go out, try and collect all these things. So we look at the next best way to do this, and that is to reaching out to local herbariums, which is just a pressed plant library. And so this is my color-coded nonsense map of trying to figure out where, if I got plants, or if I got the tissue, if I haven't gotten the tissue yet, and then just a picture of me going through some of the bags of tissue that they sent us. And so for some of you who don't know what an herbarium specimen looks like, it actually looks a lot like some of the pictures around this room, where it's just a sheet of a dahlia or any plant species is laid on a piece of paper that typically is very water absorbing, and then after it gets pressed for a bit and dried, people will go in and they'll position the plant exactly how they want it to look. And typically what people would use these for is you want to be able to see leaf morphologies. You want to be able to see the top side and the bottom side of leaves. And if there's any flowers, you want to be able to see the flowers. Primarily because some of these specimens that I got are what's called a type specimen, which is what that particular species is named off of. And if anyone has any questions about any other, for example, if someone found a Dahlia coccinea, they're like, I don't know what this is, they can go back to that type specimen and see how closely related, how similar they are. And while I was putting this presentation together, I realized that one of the herbarium specimens I got was from, I think it's 1816. That's this one all the way over here. And the fact that there's still flowers and leaf tissue on here and it didn't all degrade away after almost 200 years is pretty exceptional. So for here, we took a leaf sample, which obviously some of these are very old specimens. So you can only take a small amount of tissue. And so we were able to take roughly about this size of tissue, so just like a fingernail clipping's worth of DNA, and we put that into a tube where we then are going to grind it up, sort of like if you were here for the virus talk where they do their DNA extractions. We basically put a little pestle on a drill, and I sat there for a couple hours just drilling into these tubes, trying to grind up these herbarium specimens as best as I could. And then we go through what's called a C-tab extraction, which if anyone has ever heard of like the strawberry soap DNA, where you can extract DNA from strawberries using just Dawn dish soap. It's essentially the same thing as what we do, but we just use a little bit more chemicals to try and get it a little bit cleaner. And then so this is just a tube showing the separation of this top part is all of our DNA. This middle section is all of our plant debris, so that's like our cell walls, our chloroplasts, and all that stuff. And at the bottom is the chloroform that we have to use in order to actually separate these things. And then we put it in some cold isopropyl alcohol or an ethanol to spin it down, and then we get something that looks just like a big old blob at the bottom, but that big old blob is all of our DNA that we'll use for downstream analyses and everything. So I did that for about 170 individuals across across multiple different species, multiple different replicates of each species. And sort of what I talked about earlier is how Dahlia is a very big genome. It would not be very cost effective for us to try and sequence all of these at the same depth we did our Dahlia genome. And so to do this, we did something called target-based bait capture or enrichment, where instead of looking for, another example tied back to the virus talk is how they had like eight genes. Dahlia has roughly 160,000 genes. And instead of trying to sequence all of those, we're able to look for roughly about 1,061 genes that are shared amongst all Asteraceae. So the entire flowering plants that include sunflower, lettuce, artichokes, mums, all of those. This greatly allows us to reduce the cost of sampling as well as include more samples in our study. And essentially, this is just fishing for genes. So we take all of our DNA, we put it into a PCR machine with all of our targets that we want to put in, and we're like, okay, we're going to set you in, and I'm hoping that the DNA is going to bind to this particular bait. And then we can go through a bunch of different library prep protocols to actually build this phylogeny or a family tree of cultivated Dahlia. And so I'm just going to give a preface of this next slide. It's going to be very hard to read because there's a lot of information on here. And we'll zoom in on the important part. But so this is the family tree of cultivated the Dahlia genus based off of the samples that I collected. And the really important thing that I'm glad happened on this is we have these couple non-Dahlias out here, which is very good to show me that they're not in the rest of the Dahlias because those are my outgroups. That's like Bidens, Coreopsis, and Marigolds. And if those ended up somewhere in with the rest of the Dahlias, I'd be a little bit concerned. But so the main thing I was looking at here is our main region of interest is in this big group, which is essentially where we find a couple of different Dahlia variabilis or cultivated Dahlias. Because one of my main aims for my dissertation is seeing if we can identify the potential progenitor species of cultivated Dahlia as they went through their evolutionary history, as they came from Mexico to Europe and then back to America. And so ideally, the way I'm hoping is that if we put a couple different cultivated dahlias in here, we'd be able to, they'd all fall out together, and we'd be able to see, okay, what species is closest to it? It's like what one is its cousin? And then we can work our way backwards and try and figure out the potential progenitors. So when I was looking at this, I noticed that we had a big group of cultivated dahlias over here, and then up here we have a couple different dahlias. And so that was like, well, that's not the greatest, but by looking at by actually digging through the tree, you can see there's only a couple different dahlia species that make up this huge list. And that would be Dahlia coccinea, Dahlia mollis, Dahlia sorensenii, Dahlia parvibactera, Dahlia cardiphylla, and Dahlia artopurpurea. And so from here, this also corresponds with a couple other studies that have been put out there. The most prominent one is from 1970, where they looked at the different color groups in cultivated Dahlia species, because there's a handful of dahlias that will make white, pink, and purple flowers, and then only like 3 or 4 dahlia species that will make a red, orange, and yellow. And that's based on what types of anthocyanins and flavonoids are present in the actual dahlia. And then based off of the show out right down the hall, you can see that cultivated dahlia spans this entire color spectrum that's found in the Dahlia genus. So one of the prevailing hypotheses is that a group 1 dahlia, so the white, pink, and purples, had to have hybridized somewhere in its evolutionary history with a group 2 making the red, orange, and yellows. So that's what led up to this thought of, okay, can we find the progenitors, but also can we use the other species around it to actually try and introduce new crops? And so basically we can do something called a Kamer analysis, which is essentially we take all of our reads that we get, and we're going to break it down into smaller sections. So it's like if we're reading a sentence, we're going to make it into smaller words that are more common amongst everything. And so from here, we can take all of our reads, so all 120 samples that I have, and turn them into what is called a Kamer, basically a Kamer text file. And we're going to see, okay, what string of A, T, C's, and G's is only found in this one particular species, and then what's found amongst all these other species. And then basically you can find a way to be like, okay, what is only specific to Dahlia coccinea? What is only specific to Dahlia sorensenii? And then we can take those and map them back to our genome. That's what I have here on the bottom. And I'll go through a little bit of what these lines are. Essentially, there's 6 different lines on here, but they all get covered up by a couple of the bigger ones. Basically, the main thing to take away from this is that this red line is all Dahlia coccinea, and you can see it on every single chromosome, which leads me to believe that Dahlia coccinea is more likely to be a progenitor than a couple of the other species. And why this is important is that we can essentially do like a embarked DNA test that you can do with your dogs on potential cultivars, where we can sequence a cultivar and be able to tell you, based off of genetic relatedness to other species, how much of that species is actually integrated into your Dahlia genome, and whether or not that plays any roles in the traits or its phenotypes that it has. So basically going with that, it basically tells us that the top 2 species based on this map is a Dahlia coccinea and a Dahlia artopurpurea, which would correspond to our hypothesis that you have a red, orange, yellow Dahlia had to hybridize with a white, pink, and purple, at least based off of these phenotypes. And Dahlia coccinea is one of the few species where it does make red, orange, and yellow flowers. And so with that, the big takeaways that I want you guys to be able to go home with is that there are 2 Dahlia genomes out there that have been assembled in the past year. The possibilities for genetic research are basically endless. It just depends on how well you design an experiment. When with that, we're able to start finding potential genes beyond the genes of interest behind your favorite Dahlia cultivar. And now we're working towards identifying the parents of cultivated Dahlia, so we may potentially introduce new traits of interest from related species. And with that, I'd like to thank Dr. Walbott, who started the project, Christine Albrecht for helping fundraise for this project, because my entire PhD and all the research here was funded by all of you, the American Dahlia Society. Martin Krall, Brad Freeman, and Jim Chewy for helping source materials, and especially all of you, because without any of this project would not have been possible. And I just have a list of all the herbarium places that I got tissue from. And with that, I'll be more than happy to take any questions.
So the sample that you got, or the first sample, you used that 1800 sample. Is that when you got that
This
sample?
one.
Yep. Yeah. So when your first sample, you discovered it had those, what was that, 8 different strains of Dahlia? Is that the Coccidia, the Dahlia?
So part of the reason that we're looking at all the other different species is in the literature we know that the species Dahlias are half of whatever cultivated Dahlia is. And so I guess to answer your question is we technically still don't know the number of copies this particular cultivar, this sample has, because we just did targeted sequencing.
Okay.
So we just did small amounts of it. We didn't do the whole thing.
Sure. So every Dahlia now is actually the previous generation is one of those original the Dahlia Coccidia, the species of Dahlia.
Yeah, from at least from right now, I think it definitely has it in its ancestry. So it definitely came from a cross that then went through a bunch of different hybridization or natural selection and people picking what flowers they wanted the best.
It's amazing you can get a 10-inch out of this little original one.
Yeah. Yes.
Based on your research, is it ever going to be possible for there to be a blue Dahlia?
So that's a great question. Based off of the actual biochemical pathways in Dahlia, no. But if you were to do something like genetically modify the plant to introduce the enzymes to digest blue, because Dahlia makes, I think it's 2 steps up from making blue, then it all gets converted into a different anthocyanin. So if you're able to introduce the 2 enzymes that actually will break down the purple into, I think it's like a couple other colors, then you could get a blue Dahlia. But not I don't think not naturally.
Okay.
Yeah.
And there are blue roses, GM rose
Yep.
in Japan. Through genetic modification.
Yeah.
And you cannot cross 2 different species, not Dahlias and roses. You can't.
Oh, no.
Or orchids and.
Nope. Yeah, because
For a while there was a
Yes, like there's a cross between a Dahlia and a daffodil.
yeah, and there's just no way that's gonna.
nope.
Now, would it? Well, how about a sunflower though?
Nope. So I don't have any pictures on here, but because Dahlia is in, like I said, in the Asteraceae, which has sunflower in it. There's 20,000 species in there. The way that these plants are able to differentiate what pollen is their pollen from, like when a bee goes to a sunflower and then goes on to a Dahlia, is if you look at it on a microscopic level, they'll have varying different bumps and shapes and everything, so that your Dahlia will only be receptive to other Dahlia pollen.
Okay.
So like you could try and cross it with other asters, but it just wouldn't work. Yeah.
Even if you manually do it.
Yep.
And it's the species barrier, if you will.
Yeah. Yes.
Is there any insight on virus resistance?
I have not done anything on that yet because my particular experimental design is based more on floral architecture and floral color. But if we set up a good experimental design, we could potentially look for disease resistance and viral resistance. Yes.
When breeding Dahlias, it seems like some crosses just don't work. They won't make seed that's viable. Is there anything in your research to explain why that phenomenon?
Not yet, but I have a couple hypotheses. One of which is based off of the chromosome architecture, so like how the types of chromosomes are inherited by that particular plant. Because based off of the stuff that I've been doing so far, the 2 genomes that are out there, they have the same exact 464 chromosomes, which makes sense because they're the same phenotypic flower. They're both large yellow flowers. And so we haven't been able to do much research into, because I guess also in some cases, if a certain chromosome isn't able to cross over and recombine with its pairs, it could cause the actual gamete or cells, pollen, to just go through apoptosis and die because it's not going to be viable.
Did you find any instances of, well, we talk about there's a Sunday sport where how did you discounted those, or did you find them or not find them?
So I didn't really look into color yet at the moment because there's been a lot of research out of Japan that does a lot of color stuff.
Yeah.
But so my next steps is to actually try and use some of the sports that we have sequenced, because those would be the best ones to be able to determine what causes the color variation, because you can just say, why is this one this color? Why is this one this color? Oh, it's just this one gene that's changed. Yeah.
I would think it'd be interesting because there's the Piketty, and then you have the dual color, the backside and the front side. It's like,
yeah, there's so many, like I said, the possibilities are endless. So what kind of experiments we could end up finding.
and dark stems.
Yep.
Yeah, the foliage and all that.
Yeah.
Yeah, the challenge is in case of qualitative traits like color, it's usually determined by not one gene, not even a handful of genes. Could be dozens of genes, and there could be an additive effect. When the genes are expressed at the same time, there is one effect, and if somehow turned off, it would be a different outcome.
Yep.
So it's a
So
very
is that
complex.
going to maybe be a form of research in the future? Let's say we want a yellow or we want a white, to see if we can get everything to come out as a white.
Yeah, there are definitely ways that you can like potentially sequence your two parent lines that you want, and we can then give you a predictive guess of like 50% of your seeds are going to be this based off of your parents, and like 20% could be this. Essentially just making a big old Punnett square if you...
And you might have 5% that's on no.
Yep. Yeah. Yes.
The idea came up about developing the green Dahlia. Do you develop the green Dahlia and no one will buy it?
Yeah.
You know, follow the market. Don't
Yep.
do diversity for its own sake.
Yeah.
Yeah, we have this internal joke in scientific community. Why do they climb Everest? Because it is a collective certainty, and you have to put years of training and all that. The answer is because it's there.
Yeah, because it's there.
So, yeah, exactly. We keep that in mind before doing an experiment.
Yeah.
We are doing it just for the sake of it.
A lot of times you got to come back out of the rabbit hole because you're like, you've been staring at this one thing for so long, and you're like, I need to step back because only like 2 people in the world are going to care about this.
Your conclusion that there are 4 pairs of chromosomes as opposed to 8 is really groundbreaking.
Yeah.
Over the last 10 or 15 years, most of the literature has said there are 8.
Yep. That was one because when I started, I thought it was going to be an octopoid based off all the literature that I was reviewing. And then when I opened up that map for the first time, I was like, I'm missing like half of what I'm supposed to have. But then I like did a bunch of different analyses, and I was like, nope, that's correct. And so I was really excited by that.
Wow. It's
Yeah.
simplifying it a little bit.
A lot. Yeah.
Those herbarium samples are gold mine. And
Yes.
amazing that you got hold of those samples is really difficult.
Yeah.
These herbarium are very, particular about not part
What
of
these
the
samples that are like here, show me.
Yeah.
Yep.
So what we thought was when Alex said that he got hold of these samples, my eyes lit up.
Are they viral?
How did you get them? Yeah.
So
Return them?
yeah, so I reached out to the herbarium curator at Auburn and said, hey, I want to try and get these particular samples. I went through a whole database and went out and found each and every single one of these. And said, hey, I want to try and use this for this particular, I want to sample it so I can do a DNA extraction. And then they reach out to their particular herbarium from where these are. Like some of these are from New York Botanical Garden. One of them is from Northern Illinois University. And then they will either mail you the sample in like a very padded envelope to make sure nothing gets broken, and then the herbarium curator will go through and actually sample it for you. So I never touched any of these samples. It was all done by herbarium lab techs or the herbarium curators, where they went out and took samples off of it. In some cases, some of the plants, they have extra packets of plant material for that particular reason, where people can go back and do genetic testings on them.
Yeah, our interest in these samples was that we wanted to see if we can detect any of the viruses there.
Oh, yes.
And especially there is a DNA sequence we found years ago that is actually integrated into the Dahlia genome. Viruses do that. Now we know that in sugarcane, in banana, in rice, these viruses inserted themselves physically into the host chromosome and kind of propagate along with the plant. So interest is to see this particular integrant we found in the cultivated Dahlia. Is it there in those? So that it gives us an idea when that integration might have happened. But yeah, we talked about that. The results were inconclusive because the DNA we got was not enough quantity to make any conclusion. But yeah, a lot of kind of forensic stuff. Yes.
If you have a Dahlia with a DNA virus in it, but it's a DNA virus that doesn't really harm the plant, will that plant be more prone to picking up additional viruses, or is it not related?
Very good question. The question is that, okay, we have this Dahlia plant with this short piece of DNA. Again, that's why we are very careful. We say that Dahlias have this integrated virus, but it's not actually a complete set of virus genome. But we found pieces of that virus here and there, distributed among those different 64 chromosomes. So chances of them coming to form a complete set of a viral genome that's infectious is very unlikely. And that's why we don't think that that integrant will really cause disease. But there are instances in case of banana that it's shown that the integrated sequences can be activated under specific conditions and cause disease. And that too only 2 types of banana when they're crossed. So it's a very unique phenomenon where a dormant virus sequence inside a host DNA can be activated to cause disease. Yeah, very unique. Yeah, in Dahlia we don't have that evidence that is happening. But that reminds me of this famous quote from the first Jurassic Park. Jeff Goldblum says, "Life finds a way." When the scientist says that we have these dinosaurs, but we make sure that they don't breed. Right. But then this mathematician says, he says, life finds a way, and that's what happens.
Zach, why don't you talk a little bit about your next steps there?
Yeah, so hopefully I graduate in the spring. That's all contingent on how fast I write my couple of manuscripts that I have to write. One of them will hopefully be done at the end of this month, maybe. We'll see. And yeah, then it's just trying to figure out what to go do from there. So I'm doing job applications and writing postdoctoral grants and stuff.
But you definitely have scientific contributions here that are very, very significant. So.
Yep, and some of these ones, when floral forms, can also potentially be used in like sunflower research, because again, we have a mutant library that we didn't have to go out and create in the span of my PhD, and some of these traits could be of high interest to sunflower production and stuff like that.
Why?
So, for an example, I know there's a couple people in my lab who works on a crop sort of related to sunflower called silphium. And it's sort of in the same boat as sunflower, where a lot of people who grow sunflowers grow it for sunflower oil and oil production. And you need to be able to have disc florets that are reproductively viable, because ray florets typically aren't. And so if you have a sunflower cultivar that's really big but it has mainly ray florets, your seed and oil production is going to be really low because you aren't making seeds. Yeah.
So from next spring, please remember we have calling Dr. Zach.
Hopefully.